Or maybe I'll just hold it. So I'm interested in attention and short-term memory, and one idea that's been very potent for people who study the kinds of things that I study can be traced back at least 75 years to Hebb. And in the same volume where he famously articulated the principles that have come to be known as long-term potentiation, he talked about a dual trace. And he had the intuition that something like protein synthesis, although it hadn't yet been, the double helix hadn't yet been characterized, but he had this intuition that some metabolic process had to take place in order for reorganization, synaptic reorganization to occur, and that would have to play out over some period of time. And so the first of Hebb's two traces was this reverberatory transient elevation of activity that would act as a placeholder for the nervous system to hold on to an active trace of the to-be-remembered information until the synaptic consolidation could happen, the reorganization could happen. And in the minds of many people then, I just did a quick calculation in my head, this was 45 years ago now, on this very campus, that Joachim Fuster and Gerrit Alexander, and more or less at the same time in Kyoto, Kubota and Nikki recorded extracellularly from the prefrontal cortex of awake behaving monkeys. In the case of Fuster and Alexander, they were also in the MD nucleus of the thalamus, and recorded this very interesting property of neurons in the prefrontal cortex, is what I'll emphasize, which was that they showed sustained activity that was elevated as they reported it throughout the delay period. While the animal knew what the response had to be, but had to withhold the response until a go signal was, or actually this was in a dummy PTA, and so like the monkey had to hold its response until the screen was raised and it could respond. Now, importantly, neither this UCLA group nor the Kyoto group attributed this sustained activity to a memory trace per se. If you read their papers, they were very careful to say that these were attentional signals. But other people who didn't do the experiments but who read their experiments thought that this looked seductively like Hebb's first trace of the dual trace. And for a long time then, there's been this idea that the way we hold information over a short period of time, if I were to give James Bisley a phone number and ask him to call my wife because I forgot something that my kids have to do at school today, and so he's walking out of the room and he's remembering to himself, 608-992-4378, which is not actually my phone number, right? But we have this intuition that there's this elevated representation of 608-992, whatever I just said, that, and indeed, if he gets out into the hallway and forgets, it's because the active trace somehow went away. Now, not every lab has it been found, in fact, that there's good evidence for sustained active trace. And so this is from Tatiana Pasternak and her student from a few years ago now, where the monkeys were remembering the direction of motion of dots that were presented in a random dot kinematogram. And the animals had to make a match, non-match discrimination after a delay about whether a test patch of motion was in the same direction or a different direction from the sample direction of motion. And what they found, these are individual cells in MT and in prefrontal cortex in the caudal third of the principal sulcus. But it's probably easier just to look at the histograms. So on average, individual units in MT that were tuned for the preferred direction of the sample showed elevated activity while the sample was on the screen, but very quickly this activity dropped to baseline. So one would be tempted to say that the memory must not be living in MT because there's no evidence of sustained activity. Similarly, the cells that are tuned for the anti-preferred direction showed a phasic response, but one that dropped off. In prefrontal cortex, the story is more or less the same. There's maybe more of a hint of sustained activity, but by the time you get to the end of the delay period, there are no neurons that showed elevated activity through the delay period in a manner that would correspond with intuitions about how a short-term memory operation ought to be sustained by the brain. So with this context then, in my group, one of the things that we've done is ask our subjects, and in our case we work with the awake-behaving college students as our experimental model. And so we have our humans, our neurologically healthy humans, look at 3 patches of stimuli. In one case, these 2 are not moving, and so there's only one direction of motion within this aperture. And so this is a load of 1 trial, and at the end of the delay period, then subjects get this dial, and they have to move it to align the dial with the remembered direction of motion. In a load of 3 trial, they get 3 consecutive presentations, 3 consecutive stimuli. In this case, it's movement in the 3 o'clock direction, this is in the 4 o'clock direction, and this is in the 8:30 direction. And at the end of the trial, then, they get the same dial, but the digit in the middle tells them whether it's the first, the second, or the third that they have to retrieve. And so this is a load manipulation where we're looking at load of 3 versus load of 1, and we'll look at the sample evoked activity, which will be painted in green on the brain I show you in the next trial, versus the delay period activity, which will be painted in red. And the people who actually did the work are Adam Regal and Steve Emmerich in particular, and then Josh LaRock did some of the follow-up studies. So these are data from these are real data from a single subject, but I'm treating it like a cartoon. So this is an inflated left hemisphere, the lateral surface and the medial surface, frontal pole, occipital pole. And so color-coded in green are the areas that show a sample evoked response. So this includes the human, so this is the calprin in this inflated brain, here's the calprin sulcus. So you can see right here the representation of the fovea, as well as a lot of lateral, ventral and lateral areas. Human MT is right about here, going up along the intraparietal sulcus into areas in the intraparietal sulcus that are often thought of as a human analog of LIP. As well as in some frontal areas. The yellow, of course, is the intersection, and so there are a lot of areas that show sustained activity during the delay that is carried over from the sample. And then there are also these areas, especially in frontal and parietal cortex, that show sustained activity during the delay when there's no longer any stimulation on the screen. Okay, so this is what the trial average time series looks like. So from these frontal areas, with a load of 1, you get an initial transient, some sustained activity across the delay period, and then a transient associated with the presentation of the probe, the recall portion at the end of the trial. And you see that there is this load sensitivity in that the signal is scaling monotonically with the number of items that our subjects have to retain. When we look in posterior areas, and this is taken from human MT, the first thing I'll point out is the scale is different. So this is 1% signal change. And so the maximum excursion in the frontal cortex, you can see, is around 0.7%. In MT, it's up around 1%. Yeah. So it's a bigger signal in the back of the brain. Nice, robust, load-sensitive stimulus-evoked response. During the delay period, the signal goes back to baseline. So this was maybe comparable to what I showed you in Pasternak's monkeys, in that in MT, there's no evidence for sustained activity across the delay. There is evidence for sustained activity across the delay in the frontal cortex, as well as in parietal areas. I'm not going to show that, but the parietal time courses look very similar to the frontal. What I'm going to do next, though, is extract a region of interest, the 500 or so voxels that are active in the front, the 500 or so voxels that are active in a posterior region, and submit this to a multivariate pattern classification. So I'm assuming for this crowd. I don't have to go into a lot of explanation of how an MBPA works. I'm happy to do so if anybody wants some clarification. But suffice it to say, what we do is we train the classifier to discriminate directions of motion in 3 canonical directions. And they're canonical for us. The subjects don't know it. They're on off diagonals, but we repeat those critical directions enough times that we can train the classifier to make that discrimination. We hold one trial out, we feed it unlabeled to the classifier, and we tell it, okay, tell us, give us your best guess about what direction of motion the subject was viewing and remembering on this trial. And the extent to which this machine learning approach is successful, we can then conclude that there is information in that area of the brain that's related to the task that the subject is doing. And so when we do that, so just to unpack this slide a little bit, there's a dot here at about 6 seconds. So 6 seconds is when the maximal excursion of the stimulus-evoked signal occurs, because this is a sluggish, cold signal that we're measuring. And so when the signal is maximal, we train the classifier on that TR, and that will be what we'll call the sample-evoked portion of the trial. And in area MT, when we train the classifier on that TR, and then slide it backwards and forwards across the entirety of the time series, we're able to reconstruct the identity of the stimulus that was being remembered on that trial at a level that's above chance until 2/3 of the way through the delay, and then there's an increase again associated with the test stimulus. Now, when we have a second classifier that we train right at the end of the delay period, and again, because this is a bold signal, we train on the 14th TR, and we can be confident that there's no response-related signal that will contaminate it, because that won't evolve until the ensuing 6 seconds, because of the low-pass filter properties of the fMRI signal. We train the classifier on this late delay, and we sweep it back and forth. And so what I'd be inclined to say is that at this course level that we can talk about a neural representation, that the neural representation of motion seems to transform from a sensory-based representation to some other, I'm not going to say what it is, but something other than sensory related by the time you get to the end of the delay period. And that there's this gradual transition from the green code to the red code. Really what's important though, the point for this slide, is that we're unable to decode from prefrontal cortex. If we just focus on sort of classical lateral prefrontal, if we go back to get these areas that are more caudal, that are sort of closer to the frontal eye fields, same thing. We're unable to perform different from chance, which is a null result, but it at least is a failure to find evidence for stimulus information in these frontal regions, and the same would be true if I showed you the parietal cortex. Now, it's important to keep in mind that successful decoding isn't in any way directly measuring the representation, right? What it's telling us is that our decoder, in this case is logistic regression, is able to find discriminable patterns that are systematically related to the 3 directions of motion that we care about. I feel comfortable, though, asserting that it has something to do with the representation because of the following properties of our classification. If we just look at these posterior areas, and we look at performance of the classifier as a function of memory load, we can just take this red subject here. So for this subject, we'll call him Joe Smith. This is his classifier performance at a load of 1, his classifier performance at a load of 2, and his classifier performance at a load of 3. And you can see that the performance of a classifier, which I'm going to say is a proxy for the fidelity of the neural representation, quite nicely predicts our psychophysical estimates of the precision of the memory, which is to say that when we fit the response data, the behavioral data, with a mixture model that tells us what the precision of the memory is, what we see is that for each of our subjects, there's different offsets for different individuals, right? But the general pattern is that for each subject, the concentration parameter, which corresponds to the precision, is highest for one item, gets lower for two, and gets lower for three. So the multivariate analysis of the neural data mirrors nicely what we know about the behavior, which is that the memories seem to be less strong as we give subjects more items to remember. Okay. So from a theoretical standpoint, we're thinking about this in the context of mechanisms that have two different labels. If you're studying vision and short-term memory for visual stimuli, this will often be referred to as sensory motor recruitment. If you're studying short-term memory for words or for the digits in a phone number, like my earlier anecdote, you might think of this as the temporary activation of long-term memory representations, right? If you really wanted to push it, right, if Joachim Fuster were here, he could make the argument, and I wouldn't disagree with him, that, you know, in a sense, perception is also a demonstration of long-term memory, right? Because we had to learn, there are many examples, with motion, for example, when Pasternak and Motion showed that if you raise a cat in an environment where there's constant flicker, that they're akinetopsic, right? So our ability to perceive motion depends on, in effect, on memory from the nervous system of early development, right, of structured stimulation that shapes how our perceptual systems work. So the idea, just in really cartoon form, and this was proposed in a purely psychological context by Nelson Cowan several years ago now, but the idea is that stimulus information comes in. If it's visual information, you can think of it as being sustained in a brief sensory store. If it's semantically representable information, then the storage perhaps happens kind of more in the cognitive part of the brain, right? Whether this is more or less a reflection of the activation of long-term memory isn't a debate I want to get into. But what I'm going to talk about now is another implication of this way of thinking about short-term memory, right? So instead of thinking about there being a box somewhere in the head, say in the prefrontal cortex, whose job is to hold information over a short period of time, if I'm going to argue that we are able to accomplish these behaviors by directing attention on sensory parts or representational parts of the brain so that there's no dedicated short-term memory store, right? But that we accomplish short-term memory through the allocation of attention to knowledge structures that existed in the brain before you walked into the lab that day. Then we also have to have some understanding of the dynamics of how we control the focus of attention, right? So if I had told James, right, to remember this phone number, and if he was clever enough to know that 608 is the area code for Madison, Wisconsin, which is the area code for my wife's cell phone, he may be thinking, as he gets outside to make the phone call, okay, what's the Madison area code? Yeah, 608. And now he has to shift his attention to the 7 digits that make up her local phone number, right? So how do we accomplish this shifting of attention among items that are all being held in short-term memory? Okay, so a way that we've tried to operationalize this is stealing a technique that Kia Nobre has used very profitably, Klaus Oberauer as well, which is that we present 2 items. I'll just focus on this. This is how we train our classifier. But we present 2 items, in this case, motion going up and to the right and motion going downward. And it's color-coded, but the colors are only helpful in that they allow us to refer to cue the subject as to whether this is the critical direction or this is the critical direction. So the subject, after a 7-second delay, gets a retro cue that says it's the red direction of motion that you're about to be tested on. And now she makes a yes-no discrimination as to whether this probe direction of motion is the same or different from what she had seen initially. Now there's going to be a second brief delay and a second retro cue. And with a probability of 0.5, that retro cue will tell her, now shift your attention to the blue direction of motion, because at the end of the trial, I'm going to show you this patch of motion, and you have to say yes or no, is this a match of what you saw here? Right? And on half of the trials, the cue would be red, which would mean you're going to get another test on this. So when we do that, what I'm going to show you now is the classifier evidence, so it's logistic regression, so essentially you can think of this as the a readout of the parameter estimates from the regression that's run through the logistic function. And so the values can vary from 0 to 1.0. And it's averaged across many trials, and so we have this effectively continuous measure. And I'm going to show you that as a function of time. And so what we're doing is testing, applying the classification to each TR as we go across the trials. There are 3 directions of motion that are critical. So one is going to be color-coded in red and one in blue. Gray is going to be our empirical baseline. So when the stimuli are first presented, we see an elevation in evidence for an active representation, these 2 directions of motion, relative to the classifier's evidence for the 3rd direction of motion, which wasn't even presented on that trial. Right? So this is our empirical baseline. The extent to which these stay above this is what I'm going to take as evidence for an active representation. Now, at this point in the trial, the subject is going to be cued that the red direction of motion is what's critical. And what we see, not surprisingly, is that evidence for the red direction of motion is now elevated in the brain relative to evidence for the blue direction of motion. But what we didn't expect to see is that, in fact, evidence for an active representation of the unattended direction drops to baseline. So in our hands, we're no longer able to find evidence that the information that is in short-term memory, but outside of the focus of attention, that information is stored in a manner that's any different from information about the 3rd direction that wasn't even presented on that trial. Right? And if subsequently the subject is cued that now the blue direction of motion is critical, we see this crossover, so that now this is the effect, right? When the blue information is critical, the subjects had no problem bringing it back. In terms of this, I'm showing you data from their brains, but also behaviorally, they had no problems switching their attention to the previously unattended information. So how do we make sense out of this phenomenon here is the question that I'm going to be puzzling over. So this is a null result, right? The best we can do, in this case, we calculated Bayes factors. And so the Bayes factor that for this evidence for an active representation, the attended information is 9.7, right, which is whopping. The Bayes factor for the likelihood that the unattended memory item is higher than the baseline is actually below 1, right? So from a Bayesian perspective, it's 5 times more likely that the baseline is higher than the unattended in this case. We've replicated this several times with, let me just finish this thought here, working at the category level where 2 items are active, one gets retrocued, the other one drops the baseline. This is with fMRI. This is with EEG, where 2 items are in memory, one gets cued, the other one drops the baseline. If it gets cued again, it stays elevated. So it's a robust null result that we've replicated over several iterations.
Is it MT or EFC?
So, yeah, so this is MT, this is whole brain, fMRI, and this is whole cap EEG, spectrally transformed EEG. Yeah, just.
So in the previous slide, by the end of the trial, the classifier has strong evidence for all 3 motion directions. Do you think that the sort of neural patterns in the region contain superimposed sort of representations of all three directions?
Well, so I think what happens is that there's something like a contextual reinstatement. So whenever we present a probe, we see evidence for everything, including the unintended or the irrelevant information coming back. It's not unlike what Sean Collin recorded in that original the science paper where Collin and Norman and their colleagues, right, showed this sort of reinstatement even of irrelevant information. And so this is, it's transient, right, and it drops back down. And then this is at the end of the trial when they're pushing a button and, you know, God knows what else is happening at that point.
My confusion is more that the classifier has been trained to distinguish these three specific directions from each other. So
Correct.
it's that there's differences.
Yeah.
And then how could it think that all three are?
Well, so there are three classifiers, right? So there's, you know, 17 degrees of motion versus not 17 degrees of motion, logistic regression. There's 135 degrees of motion versus not 135 degrees, and there's 288 degrees versus not 288. And so each of these are independent then outputs of that individual classifier that's dedicated to that one stimulus type.
Okay,
Does that make sense? Yeah, okay. Yeah, I'm sorry if that wasn't clear. Right. Okay, so something akin to, I'm going to call this selection, has happened when the subjects are cued that it's, you know, 17 degrees and not 288 degrees that you are going to be tested on in 8 seconds time. And so let's turn our attention then to try to understand the dynamics of this intentional selection that's happening here. And this is work that Jason Samaha has carried out, and it got published earlier this year. And as the title indicates, it's going to take us into the world of EEG and the alpha band. So the task that Jason used, and this will look familiar, some of the flavor of this task will look familiar to anybody who's familiar with Hawk-Wan Lau's work. Jason is kind of a closet science stalker. You need to know this. And so what this is temporal prediction, where in this case it's a yellow cue tells the subject with 100% validity that in 650 milliseconds this Gabor is going to come up. It's backward masked. They make a forced choice. Is it oriented to the left or to the right? And then they also make a confidence rating later on. If it's a green, then they know with 100% validity that it'll be a 1400 millisecond delay, and then they'll see this oriented Gabor, right? There are also an equal number of trials in which an uninformative blue cue is presented. And so this blue cue just says there's got to be a Gabor, but you don't know whether it's going to be 650 milliseconds or 875 milliseconds or 1,125 milliseconds or 1,400 milliseconds. So what that's going to enable us to do, right, is to compare the activity on these perfectly matched conditions, where the only difference is that here the subject knows when to expect the target, and here they don't. And of course we counterbalance across subjects with the colors so that that wouldn't be a concern. So in behavior, we see that having advanced knowledge of when the target will appear helps your performance. This is the predictive versus the unpredicted condition. These are the means here. Helps you with a short delay, not with the long delay. It turns out the way we designed the experiment, we didn't take into account the hazard rate. And 1,400 milliseconds is the longest delay that there was. So even in the uninformative condition, if you've waited around for longer than 1,100 milliseconds and the target hasn't come yet, then you know when it's going to come. And so we think that that's what just saturated the effect. It also helps subjective performance. So subjects are more confident on the predictive than the unpredictive. And again, only for the short, not for the long. These are the behavioral data, just replicating the effect, but now this is behavior collected well. Jason also recorded the EEG. So again, we see a benefit for, in the short delay, for both objective and subjective performance. when he looked at the EEG in the back of the brain, it was dominated by energy in the alpha band. So this is right around, this is an average, right around 10 hertz. And so what he did was he bandpass filtered the data to 10 hertz, and then plotted these bandpass filtered ERPs. So this won't be a spectrally transformed. This will be just the bandpass filtered waveform. And the interesting thing that he saw was that, so this is superimposed unpredictive and predictive, right, over our 15 subjects, however many subjects. What he saw was that whereas there's clear evidence for kind of a phase reset at the beginning with the cue and nice locking through the beginning of this 650 millisecond delay, at around 100 milliseconds before the target was going to come up, there's, depending on how you like to view things, there's either a change in the frequency or a phase shift, right? Such that there's a reliable difference by the time the target hits between the predictive and the unpredictive condition. And the long delay, I'm showing it for completeness sake. Remember, there wasn't a behavioral effect. There's some suggestion, though, that people's brains were treating the predictive differently than the unpredictive, but again, because of the way we designed the task, we're not able to make any strong statements about that. When we look at this more carefully, we think that this is a gradual shift in the frequency. And furthermore, when we look at individual differences, it turns out that there's considerable variability. If I were to take each of you into a local EEG lab and just have you do 30 seconds of eyes closed and eyes open and eyes closed and eyes open, your peak alpha frequency would vary considerably from individual to individual. A lot of people have done work to suggest that this is stable and trait-like, so that if we brought you back again tonight and then brought you back in 3 weeks, if you're an 11.1 hertz cycler, you'll come in at around 11.1 hertz with some noise, but it's a fairly stable trait. What we found is that in these predictive, with these predictive cues, that the fast cyclers tended to slow down. So this is predictive minus unpredictive peak alpha frequency. And that the slow cyclers tended to speed up. And it's not a regression to the mean because this was acquired independently, right? And so instead of a regression to the mean, it's actually a deviation, a systematic deviation in one direction or another, the direction and the magnitude of which is predicted by what your trait-like resting peak alpha frequency is. So frequency sliding, right? What's up with that? Just as a quick aside, there was no evidence, this is looking at power, there was no evidence that changes in power were in any way helpful in explaining these effects. They didn't differ across conditions. So one idea that you may hear about tomorrow from Brad Wojtek, but in any event, his postdoc, Eric Peterson, has been doing some really nice work, simulations with single simulated leaky integrator neurons. And what Eric has shown is that these individual neurons, that the output gain is sensitive to the electrical milieu, in that if the cell is experiencing a low frequency oscillation, somewhere below 20 hertz, that fluctuations in that oscillation will modulate the gain. So a slower oscillation is associated with higher gain in the output of that unit, and at the population level, what that translates into is essentially a higher rate of information transmission from the population, from the network, as a function of the field level oscillation that the cells are experiencing relative to no oscillation. And so here's just an example from his simulations of the performance of these simulated cells with no oscillation and with a 10 hertz oscillation. And so one possibility is that, and the idea that I'm going to push in an agnostic sense, is that the way that the cognitive system implements visual attentional control, one way, is to hijack this physiological structure in how the visual system works, which is to say it oscillates at around 10 hertz. And a more precise mechanism then for carrying out this hijacking may be to subtly shift the frequency of the oscillation so as to do something like tweak the gain characteristics of the area that's the site of potential control. Really quickly, this is from Marcello Massimini's group. This is Mario Rosanova. They delivered TMS to blindfolded subjects who were resting to the area 19 in occipital lobe, area 7 in the parietal cortex, area 6 in the frontal cortex. And what they found was that the TMS evoked, the spectral TMS evoked response was systematically different, as though if you think about an array of bells or whatever that instrument is, where a xylophone, right? The low tones are in the occipital lobe, and you get higher and higher tones as you go from caudal to rostral in the brain. So there may be something just fundamental about this sort of 10 hertz range that's fundamental to how the visual system works. So just to push that idea, is there a question? That might have been myself here. To push that idea, Jason did another study looking at, taking advantage of this property that there are stable individual differences in the frequency of alpha band cycling. And he designed a gap detection task where there is 90 milliseconds of stimulus, but in some cases, this square wave of 90 milliseconds is interrupted by a 10 millisecond gap. And here's a 100 millisecond square wave pulse of stimulus. It's a dim dot. It's either on the screen for 100 milliseconds or it's on the screen for 40 milliseconds, 20 milliseconds of gap, and 40 more milliseconds. Right? So what we're doing is just gradually increasing the width of this gap in time and asking what is the amount of separation that subjects need before they can detect the difference between an unbroken pulse of light and an interrupted gapped pulse of light. And there are individual differences, as you might imagine. So this is the group aggregate data. People are pretty good at identifying the unbroken flashes as unbroken flashes, and not surprising. So James, this is what this reviewer 5 got hung up on, right? Couldn't wrap his or her brain around the notion that with two alternative forced choice, the chance should be at 50% and not at 0. So they forced us to pull apart the two trial conditions to show that in fact you get this phenomenon of really poor performance at the brief gaps that nobody can detect. And just about everybody then is doing fine at the 50 millisecond gaps, right? And they get better as you make the task easier. There are individual differences in performance, which you can tell by the slope of these psychometric functions, right? So these are the psychometric functions that show the proportion of correct responses that goes to 0.5, not to 0, because it's correct responses as a function of the gap duration. And so the slope of this line tells you where any individual 75% threshold is. And what Jason's going to do is just regress the estimated threshold against the peak alpha for each of these subjects. And so this is just showing you that here, for example, is somebody who's relatively slow. They're at around 8 hertz. This is a test retest, the two colors. And here's somebody who's at around 10 hertz, and here's somebody who is up at 12 hertz maybe, right? And so when he does that regression, he finds between subjects that sure enough, the faster cyclers have a lower gap detection threshold, and the slower cyclers have a higher gap detection threshold. And that's true whether you estimated their peak alpha with the sort of standard, the Berger 1927, eyes open close method. This is the Michael X. Cohen 2015. You take the Hilbert transform of the 200 milliseconds prior to the stimulus, and the Hilbert transform gives you the the phase angle, and you take the first derivative of the phase angle, that you can read out frequency from that. So this is a measure of instantaneous frequency, and you can see that the effect is robust across those different ways of estimating alpha. Another thing that's really neat that we're still not quite sure what to do with, but we have some ideas, is that if you look within subject on a trial-by-trial basis, there's going to be some jitter on a trial-by-trial basis in the frequency of your alpha band cycling. Now, this is not a typo. This is true. This is 10.52 hertz and 10.54 hertz. So this is average across everybody. But within subject, when an individual's cycling slightly faster, she's doing better than when she's cycling on the trials when she's cycling fast, slightly slower. And that seems to be superimposed on this lower frequency fluctuation, right? Because you see it here for these 150 milliseconds. You don't see it for the 152. This is kind of 4 hertz range. And there's a guy named Feeblecorn who did some work with, he's in Sabina Kastner's group now, Ian Feeblecorn. And he's published some data, both psychophysical data and now physiology in a monkey, that shows good evidence for a 4 hertz sort of sampling rate that humans and monkeys are, even when they're holding their attention on one location, that they're sampling at this low frequency. They're sampling different areas in the visual scene. And so it could be that this sort of envelope over which this higher frequency thing is happening is related to something about these nested oscillators. Okay. Right. So I made this point already, right? So the cognitive system may hijack alpha band dynamics in order to implement attention in short-term memory. Can I show you any evidence for that? So let's try to get this back to attention in short-term memory. So this is Jason Samaha with Tommy Sprague, who just got his Ph.D. with John Sorensis and is now at NYU with Clay Curtis. And this is a result of a study that came out of Brain Camp at Santa Barbara last summer, where Jason was working with some folks who taught him how to use this inverted encoding modeling technique. And so publicly available data on the web, where a cue, in this case, the subject's cue is red, and so the red means that this is the aperture where the stimulus will appear. It's either going to be an X or a T, and it will be backward masked. So it's a classic Posner-style covert attention task, where your RT is much faster. These are individual subjects, where your RT is much faster when the target comes up in the attended versus an unattended location, right? This is the this is the I'm trying to think of the name of the software. I guess it doesn't matter. This is a time frequency representation, right? So here's frequency, 30 hertz, 5 hertz. This is time during the trial. And so you can see that both in the unattended and the attended hemisphere, there's this initial, what's been called alpha desynchronization. And then over the period of time when attention is covertly focused on one location, there's this asymmetry with higher alpha band power in the ipsilateral versus the contralateral hemisphere. Ipsi or contralateral to the attended visual field. And here's the difference. So this is a fairly standard result. When Jason applied MVPA, he found that only in the alpha band and not at lower or higher frequencies was he able to decode the location of covert attention. So then he bandpass filtered the data again in this range. And now he applies this forward encoding modeling approach, inverted encoding modeling, where in a nutshell, you determine what's called the population receptive field for each electrode. You can do this with fMRI as well, with individual voxels. So you just take one electrode. What is its response to attention to this location, this location, this location, this, right? So you get what amounts to a tuning curve for that electrode. You then regress the properties of each of the electrodes against this basis set of, in this case, it's rectified sinusoids that are located that span the entire 360 degrees of space. It turns out the shape of your basis functions doesn't really matter. You can do it with delta functions as well. And the point is that in doing, it's a dimensionality reduction approach that allows you to reconstruct the reconstruction of the neural representation. So once you have your model set up, your forward model set up, you can feed the model data that it's never seen before, and it will generate a reconstruction of the precision of the representation. So here is a reconstruction from Jason's data of the tuning of the system that's centered on 0, which is the targeted location. So his reconstruction of using alpha, he does as good a job at at each of the 6 locations at constructing these forward encoding models. And so then the question you can start to ask is, well, what about the temporal evolution of this, right? So over time, what is the temporal evolution of the neural representation of a covertly attended location in space? So here at this one narrow slice in time, we have this estimate of the neural representation, right? And if we color code it redundantly so that the peaks of the representation are red and the flanks are yellow, right? And we also generate a estimate of the reconstruction going back through time. And then if we, figuratively speaking, fly up in a helicopter and look down on this from the top, you can see something like this, where these are you're looking straight down on this, essentially on this mountain range, right? And so these are the channel tuning functions, the reconstructions of the representation of a location in space evolving through time. Here's when the cue was presented. And initially there is a representation of the cued location that's probably directly related to the fact that there's energy on the screen. But pretty quickly thereafter, you can see that the visual system develops this representation of the remembered location, holds on to it throughout the delay period. Now, I want to go back just for a second and talk again about the scalp level EEG. And remember, I made this point about how we saw bilateral desynchronization initially, and then gradually this asymmetric elevation in alpha band power. This is something that's been seen many, many times. And Gregor Toots' group, for example, has argued that this dynamic sort of stereotyped bilateral desynchronization followed by this gradual asymmetry can be associated with 2 discrete cognitive processes. That the suppression is associated with the shift of attention, and that the maintenance period then of sustained covert attention is associated with this asymmetric pattern of alpha band enhancement, which is a good story. These are our data, and we can see the same pattern, right? That this would be suppression and this would be enhancement associated with shifting and associated with maintenance. The problem is when we line up our multivariate analysis versus what amounts to a univariate sort of visual inspection approach, right? The time course of our multivariate channel reconstruction does not in any way accord with what you would infer from looking at the data on the scalp, right? So the data on the scalp suggests that there's this shifting that deploys over the first 800 milliseconds, and yet by 450 milliseconds, the multivariate analysis tells us that the system is already representing the target location. And so how does it make sense to say that the subject is still shifting if their brain is already representing the target location? And in fact, behaviorally, right, we know that we can see attentional effects, right? When you have an endogenous shift of attention, you would expect within 300, 400 milliseconds to start to see benefits from that endogenous cue. And so it doesn't make sense really to think that the shift is deploying over this long period of time. I'll get to you in a second. And then just as a second way of looking at this, we also did a cross-temporal classification where we trained on the the alpha bandpass, the scalp distribution of alpha power, and we test on not only the same time point, but on different time points. So to what extent do we get cross-temporal classification? The extent to which you get cross-temporal classification, right? The extent to which what the classifier learns at time 1,500 milliseconds can also be useful for decoding 2,000 or 500 suggests that there's a stable neural representation. And we see that there's this stable neural representation similar to the forward encoding model. The stable neural representation is already in place at cortex milliseconds. And so it's possible that there's some kind of multiplex thing going on where what you see at the scalp is a different process than what you see here. I'm more inclined to think that what you see at the scalp is no more informative than is visual inspection of bold signal intensity, right? That when you look at bold signal intensity, you say, oh look, it scales monotonically with load, but it turns out that's not very informative in terms of what that brain area is actually doing, whether it's representing the stimulus or not. Question?
So is it true that the target is represented also just
Stimulus evoked because the cue is on the screen, it's telling the subject, right? So this I'm not going to call covert attention, but this I feel comfortable calling covert attention. Yeah. And actually you see the same thing, so is this relevant for working memory? It is, because Josh Foster and Edo's group has done exactly the same thing, and here you can see a lot more subjects, in fact, and they more carefully actually used a very subtle cueing technique that removed any sensory component. And you can see that their channel reconstruction very nicely picks up even earlier than ours did, right? On the order of 250, 300 milliseconds. And so here's the spatial short-term memory from Ed O's group. This is covert attention from Jason Samaha. And so based on this similarity, do we want to endorse this idea that short-term memory may be nothing more than selective attention extended in time? And I feel comfortable saying that I think that's a reasonable thing to do. Do we want to say that visual working memory is nothing more than selective attention extended in time? There I'm going to say no, and the reason for that is, remember that we already looked at this dissociation between the focus of attention and items that are in short-term memory but not in the focus of attention, right? So there must be some mechanism that's holding this unattended information in memory when it's no longer in the focus of attention. And so for the last little bit of time that I have here, I'm going to tell you about some work in progress that's trying to get at the question of how can this be, right? How can it be that information is held in short-term memory, but we can't find evidence for a sustained trace? And the idea that we're going to work with is that perhaps short-term memory at its foundation is no different than long-term memory, right? Maybe Head got this part wrong, which is to say that Head didn't know about short-term plasticity, right? Head didn't know about spike time-dependent plasticity, which we know on the order of milliseconds will determine whether a synapse is strengthened or weakened, depending on the timing of the incoming volley versus the discharge of the cell, right? So the idea then is one that has been worked out computationally by Misha Sodix and his colleagues, and it goes something like this. If you have structured information that's coming in from the environment, that structured information can be encoded in neural activity in a bank of filters. And later, if you just blast a sweep of white noise through this structured filter, right, this bank of filters, the output of this bank of filters will reflect the information that it previously encoded when it was given the structured information. And then you can use the match filter principle or something like that to actually solve the short-term memory problem. Right? So the idea then, just to be really concrete, here's your bank of filters, right? These are individual neurons. In my cartoon, their activity level is really high and really low based on the inputs. And then later on, it's no longer the activity level, but it's the synaptic strength that's transiently encoded. And so then if you feed any kind of information, any kind of energy into the network, even if it's not structured, the output will reflect this difference in synaptic waves. And so what Nate Rose, who just got a job at Notre Dame, who was at Australian Catholic University in Melbourne, doing a sort of a weird second postdoc, but he has a real job now at Notre Dame. He's starting in the fall. I'm very happy for him. There are real jobs in Australia, don't get me wrong. This one didn't happen to be a real job. So we did fMRI to train the classifier about the patterns associated with the directions of motion, associated with faces, and associated with words. So this is kind of a coarse category level decoding. We did the same two-step working memory, so a face and a word. The subject is cued that the face is going to be critical, and now she makes a yes-no discrimination about the identity of the face. Now, on 50% of the trial, she's cued that it's the word. And in this case, because Frankenstein rhymes with asinine, you would endorse this as a phonological match, right? Whereas if this had been Dracula, you would reject it because Dracula doesn't rhyme with asinine. So it's phonology versus faces versus direction of motion. And again, we have then an attended and unattended memory item. We deliver TMS, and we use TMS just as a brute force way of introducing energy into the system. So this is just convincing you by showing you that the TMS evoked response, and this is a running average, so I'm accumulating an average of the TMS evoked response. Each little hiccup that you see in the movie is the addition of another pulse. And so this is just to persuade you, right, and these are microvolts, that the TMS evoked response that we get is a reasonably physiological one that we can make sense out of. And so what we do then is we present subjects with two items. They're both in memory. One of them is cued as being relevant, and our classifier tells us that, and this is the classification of the EEG, that once one of them is cued as relevant, the other one drops to baseline. So what I'm showing you here is classifier performance is the area under the ROC. So 50% is the diagonal in the ROC. When we deliver a pulse of TMS, what we see is this transient reactivation of the multivariate evidence for the unattended memory item. And this was done when the unattended item was a face. We tried to hit a face-related area. When the unattended item was phonology, we hit a phonology-related area. When Nate redid the study with another 6 subjects to convince ourselves it was real, he picked this area in the rostral part of the intraparietal sulcus, and he hit this area for every stimulus type, reasoning that if this is truly sort of a top-down attentional signal, then if we goose the source of attentional control, it shouldn't matter, right? It shouldn't be the case that we have to just hit the cortical area, because the TMS signal, of course, is going to be propagated to many brain areas. And if it's differentially propagated to the brain area that is encoding information that's relevant on that trial, perhaps we'll see a difference, right? So this is a replication study. The conventions are more or less the same, except now we're anchoring the data to the time of the delivery of TMS, and we jittered the time of delivery of TMS relative to stimulus presentation. So now stimuli were presented over this range of times, right? The cue appeared over this range of times. There's active representation of the attended, not of the unattended. TMS is delivered for a brief period now. We can recover multivariate evidence for what I'm going to call an active representation of unattended memory item. It's contextually specific in that later in the trial, they get a second cue, but now they know that the red stimulus will can only be tested, and the blue stimulus can't be tested on this trial because the cues are 100% valid. And in that case, we don't get this reanimation effect. Right? I call it the Lazarus effect. My students want to call it the Frankenstein effect. So that's kind of a generational thing. We haven't worked that out yet. The other thing I would emphasize is that these are, we're looking at the ROC, right? And so implied in this is that the extent to which the performance in the ROC is above 50% means that the false alarm rate is is low, right, to a reliable extent. And so what that means is that I can't show you the data because of the way that they were calculated, but what that means is that the representation of, let's say, faces, when faces are not presented on that trial, does not increase. So it's specific to the information that's in memory and specific to the relevance of that information for performance on the upcoming probe. Right, so as Marco told you, I'm a few hours down the coast this year in the Cognitive Science Building, which is right about here, working with Brad Wojtek's group. I'm sure Brad tomorrow will tell you about some of this really cool electrocorticography that they've done. Suffice it to say that what I'm hoping to learn this year is how to do electrocorticography in awake-behaving humans to see if, as we get closer to the action, right, because we're recording from the cortical surface, will that maybe give us more insight about the mechanisms by which information that is in short-term memory but not in the focus of attention gets retained across this brief period when it's relevant for behavior. And thank you for your attention.
Any questions?
So I have a question about the last experiment.
Yeah.
Probably, I don't know if you know this, but what is your intuition about this? So for example, if you activate, let's say, face area, and then switch the subject and present face instead of other
Right.
would that increase performance?
Yeah, so we haven't done that. What we've done that may start to get at this is we include some catch trials where the subject will be probed on unattended information. So if you're given A and B and you're told A is relevant, then if you get B as a probe, you have to reject it because A is the only relevant one. We can't do that in these studies because if we do that, then that means that our cues aren't 100% valid and that spoils the whole conceit of the experiment. But in this separate experiment, we include those catch trials and we deliver TMS with different latencies relative to when that probe comes up. And are they more likely to false alarm on B when we think that we've activated the representation of B with TMS? And the answer is invite me back next year because we're in the middle of that experiment now, but I really hope it works. Yeah, Jesse.
So as we experience the world in a moment of time, there are many experiences that are happening, and then only as we move forward, only a subset will stay in the focus of our attention. And
Sure.
it seems like everything that sort of recently happened is something that you could potentially retrieve, and you're saying that the short-term memory component that's not part of the focus of attention, it's the spike time component, plasticity, or some kind of fast-acting
Right.
synaptic change that makes that accessible. But is there a tagging mechanism that sort of says, of the things that recently happened, these are the ones that I might later be tested on, I don't have the resources now?
I think that that's part of what parietal cortex does for you. And so when people talk about a salience map, right, and maybe some frontal too, right, that we know that in what I'll call the frontal parietal manifold, part of what that network is doing is keeping track of your recent history. And you can, and in fact, James was telling me about some really nice data from his lab where you can recover information from groups of neurons about what happened in the previous trial. And we have evidence that's consistent with that idea as well. So I think that what makes this different from episodic memory for where I found the drinking fountain right before this talk, right, which can't be in my short-term memory anymore, must have been hippocampally, mediated because if I were HM, I wouldn't remember what I did right before the beginning of this talk. So the difference there is that that's where you need this sort of long-term memory engine that will lay down those traces. And what then individuates, incidental things that happened 10 minutes ago from the 5 things that I still have in short-term memory but are not in my focus of attention is probably where your salience map is pointing to, it's not active, but if you need it, right, here are the things that are relevant on this trial. Yeah?
Yeah, I
come from a different world, but some of the things that you showed that stimulated me. And I know from my own experience, both as a teacher and a researcher, that disruption, interference, destroys memory better than anything else.
Oh, sure.
I always tell my students, you go out and I'll talk about it.
There's no such thing as decay, right?
So you showed a sequence of events where there was activation from an EEG point of view, followed by deactivation.
Right.
Right? The deactivation means it was increased alpha, I presume.
No, all that meant was that our ability to discriminate patterns in the data that suggested an active representation of the stimulus dropped off. So the multivariate evidence for a multivariate representation was no longer there, but that can be independent of what the sort of local level of physiological activity is. So
do you look at those are task situations?
Yeah.
And do you actually look at performance or the function of the patterns that you see? Because if it is an increase in following the correct response, that actually blocks disruption.
Right. So again, what we're looking at is the pattern of alpha band power at different at this array of electrodes and the changes in that pattern of power. And so in principle, that can happen independent of whether one electrode's alpha is increasing or decreasing. So one thing I would point to is early in the talk where I showed that with fMRI, not with EEG, but with fMRI, that our ability to decode stimulus information dropped off systematically with the number of items that subjects held in mind, and that neural, that loss of neural precision, if you will, predicted what the behavioral estimate of the psychological precision was, in that your psychological precision is lower when you're only 3 versus 1. And that happens independent of whether, in fact, it happened in this area of the brain that was sort of dark and cold, right? Where there was no difference in the signal relative to baseline. And yet, we were able to find that really exquisite variation in multivariate stimulus representation, which you would never know if you were just evaluating the whole signal intensity, which is analogous to saying, which you would never know if you just eyeballed the alpha band power. Yeah.
I think it's a really beautiful and you presented it today, and really stimulating. And one thing that actually just, as I was going to the end of the talk, thinking about the early experiments. So one thing about the EEG was that you can encode the information by doing a machine learning approach, and that turned out to look very different from just looking at the frequency.
Right.
Would that be an argument to motivate going back to fMRI experiment that way? So the number you saw that in prefrontal, you cannot decode the content. Was because you probably took the ROI based on the activation that you did there.
Right.
What if you just run the whole...
So when we do a spotlight...
well, we just take the whole frontal parietal as a big ROI,
Yeah.
run feature selection, it was still not...
Fails. And in fact, my graduate student, Adam McGall, went so far as to go in and iteratively remove features one by one and look at what happens to your classification, brute force, right? And as he iteratively removed everything in the frontal parietal, it had no effect on classification. As soon as he started to do that in V1 or an MT, then performance just fell off. Yeah.
So in frontal parietal, you can find motion selective patches, they just don't show.
You can find motion selective when there's motion on the screen.
Yeah.
You're driving the whole brain.
And it just drops.
Yeah. And Marty Serino, a lot of people have shown that, right? What's different is that during the delay, do you see sustained representation of that motion?
I was just wondering if it's not an issue that you're not sufficiently training your class.
No, and so the other thing that we did, another sort of subtlety is in an early study that Adam did, we had the subjects making a speed or a direction discrimination. But they didn't know until halfway through the trial which it was going to be. And so what we found was that during the first half of the trial, we can decode direction of motion. Once the subject is cued that it's a speed trial, then we can no longer decode direction of motion. If we instead train the classifier to tell us, is it a speed trial or is it a motion trial, right? So what kind of trial is it? It's a chance in prefrontal cortex. As soon as the cue comes up that tells the subject what kind of trial it is, frontal cortex now knows and is telling you this is a speed trial, right? So it has that more abstracted sort of task set level information. It doesn't have, in our hands, it doesn't have the stimulus specific information. I think we're over time.
Yeah, thank you.
Thank you.